Welcome to the O-Levels Hub. It's O-Levels Biology 5090 lesson, Chapter 4, Biological Molecules. Chemical elements. Most of the molecules in living organisms fall into three categories: carbohydrates, proteins, and lipids. These all contain carbon and so are described as organic molecules. Molecule, carbohydrate. Chemical elements are carbon, oxygen, and hydrogen. Protein molecule contain carbon, oxygen, hydrogen, and nitrogen, and some contain some small amounts of other elements such as sulfur. Lipids. They contain carbon, oxygen, and hydrogen, but note that lipids contain less oxygen than carbohydrates. Carbohydrates. Long chains of simple sugars are known as carbohydrates. Glucose is a simple sugar, which is a monosaccharide. When two glucose molecules join together, the maltose is formed, which is a disaccharide. When a lots of glucose molecules join together, starch, glycogen, or cellulose can form, which is a polysaccharide. Lipids. Fats. Most fats, means lipids in the body, are made up of triglycerides. Their basic unit is 1 glycerol and 3 fatty acids. The fatty acids are vary in size and structure. Lipids are divided into fats, solids at room temperature, and oils, liquids at room temperature. Proteins. Long chains of amino acids are known as proteins. There are about 20 different amino acids. They all contain the same basic structure, but the R group is different for each one. When amino acids are joined together, a protein is formed. The amino acids can be arranged in any order, resulting in hundreds of thousands of different proteins. Even a small difference in the order of the amino acid results in a different protein being formed. We will later discuss it in the DNA structure chapter. On the right side, there is a general structure of amino acids. Proteins. How your body uses amino acids as building blocks. Amino acids are these colorful balls, and the peptide bonds are formed between them, and then a protein is formed. Chemical test. Test for glucose, which is also known as a reducing sugar test. Add Benedict solution into the sample solution in test tube. Heat it at 60 to 70 degrees Celsius in water bath for 5 minutes. Take test tube out of water bath and observe the color. A positive test will show a color change from blue to orange or brick red. Test for protein. Add drops of Biuret solution to the food sample. A positive test will show a color change from blue to violet, means purple. Here are the diagrams on the right side. Chemical test. Test for lipids, which is also known as ethanol emulsion test. Food sample is mixed with 2 cm cube of ethanol and shaken. The ethanol is added to an equal volume of cold water. A positive test will show a cloudy emulsion forming. A cloudy emulsion means just like a cloud formation of white color, milky, on the top of the liquid. Test for starch using iodine. We can use iodine to test for the presence or absence of starch in the food sample. Add drops of iodine solution to the food sample. A positive test will show a color change from orange-brown to blue-black. For example, the potato is tested for the starch. It is peeled off, and then we have put some drops of iodine on it, and the potato contains starch, so the iodine changes color from brown to blue-black.